how do we represent the sequence of amino acids for primary structure? Use the three-letter code. THR, PRO, GLU, GLU. Okay? That is a primary sequence. We're not saying anything about how that sequence then interacts with some other part of the sequence. We're just saying this is the sequence of amino acids. That's it. How else could we represent that sequence? One-letter codes. How else? Full name. How else? Structure. Okay. All of those are valid representations for the primary sequence, okay, or the primary structure. It's just the list of the amino acids. Okay? And yes, this change is important, and I do find it a bit odd that they're then going through and saying, well, look, if we just make that change, all of a sudden this stuff happens. Well, why is this stuff happening? It's not just that I changed out that amino acid. It's that when I change out that amino acid, that changes the secondary structure, and the tertiary structure, and the quaternary structure. That changes the physiological stuff that I'm looking at. Okay? So it's a bit nuanced to say, oh, primary structure is super important, because if we make a change, now everything flips out. That change has a trickle effect down to everything else behind it. Okay? And that's the part that we will talk about today, looking at then what happens after our primary structure. Okay? And where we kind of ended was looking at a single amino acid, okay, which is our structure in the middle here. So we've got our double bond oxygen, our carbonyl. Black was a horrible color there. Okay. We have the backbone of our amino acid. So if we have the carbonyl up here, that's coming from which part? The amino or the acid part? The acid part. Okay. Notice there is a nitrogen there, but that nitrogen is coming from the amino end of the next amino acid. Okay. So that's the acid portion of our amino acid, of our central amino acid, with its backbone, that then terminates in the, again, blue was a bad color, the amino end of our acid, of our amino acid. Okay. We talked about the peptide bond already. The peptide bond shows up in two places. Okay. We've got one down kind of at that bottom part that I'm trying to show in green. We've got one up at the top. Okay. What did we say was an interesting feature of that peptide bond? What's that? It can somewhat rotate. Why is it limited? Not quite. It's resonance coming from the nitrogen. The nitrogen has a lone pair. They can do resonance into the carbon of your carbonyl. That makes a double bond. Double bonds can't rotate. Okay. So, and Mark, where you were going was probably referencing these guys, and that's where we have to be careful, because the peptide bond is significantly different than the backbone bonds, I guess, of your amino acid. Okay. And the peptide bond is the one that really has a hard time rotating. Okay. So it can, and it will, but it is relatively minimal. Because of that resonance contribution, okay, that limits that rotation. Which means if that bond is now locked up and can't rotate much, how do we get motion out of our molecule or out of our protein? That's going to rely on any bond that doesn't have a pi bond, which would mean the two backbone carbon bonds of our peptide or of our amino acid. Okay? The angle at which we rotate that then gets specified as some special term, because that rotation or that angle changes our overall structure. Okay? And that's where we get that psi and, I guess we decided theta. Okay? Those are known as your Ramachandran angles, named after Ramachandran, because he was the one that went through and said, hey, these angles are important. These are what's going to dictate some of how our structure can interact and fold and do all this stuff. Okay? We can get really specific in that. We don't need to. We just need to be aware of them. Okay? The psi is officially referring to the carbon-carbon. The thetas are carbon-nitrogen. I don't think I would ever explicitly ask that, but that's part of what's going on there. Okay. We've got force and bond. What was I going for on that? What kind of bonds do we have there? Okay, we have single bonds, also known or better referred to as our sigma bonds. Okay. The force is a little bit of a weird statement there. As we make adjustments about that bond angle, what forces might be changing? It's a tricky question. Do we see anything within that peptide backbone that might have some kind of force or potential for a force? As a hint, there's an example of one drawn on the board.
Would it be the electronegativity of the oxygen?
The oxygen being exceptionally electronegative means that it becomes what charge? Partially negative. This means it can have a strong interaction with something that is partially positive. So I can generate, I've got part one of a dipole-dipole force coming out of this structure. By changing the angles, I can then bring that partial negative to bear on another positive. Is there a positive? Yes. Yes is your answer. We have the partial positive hydrogen that was connected to that nitrogen. Okay. Yes, we have a partial positive carbon. Okay. That will generate a dipole-dipole force, and that is something to consider. However, is there a stronger force than the dipole-dipole? The hydrogen bonding. So what we want to focus on are our strongest forces, and then we can move down into our weaker forces. We have that partial positive hydrogen, meaning if I adjust the bond angles around my ramichondron, I can start to bring the partial negative of my carbonyl closer or further from a partial positive from the amino or the amide nitrogen hydrogen bond. Okay. That can generate some strong interactions. Those strong attractive forces are what are primarily responsible for secondary structure. Okay. So before we advance, where are those forces originating from? It's a really vague, hard question. And theoretically, Jen and Terry should be able to answer this because they had questions in. Where are those forces originating from? Where's our partial negative originating from? Where's our partial positive originating from? And you can't tell me from the acid and the amine. Doesn't work. I want more. It's the protein backbone. Okay. Notice, where's the R group in any of those charges? R group is over here. Have we assigned any kind of charge to that R group? No. Why not? It's not relevant for secondary structure. This does not mean it's irrelevant. It just means it's not relevant for secondary. Okay? So when we're looking at our secondary structure, we are concerned about the protein backbone and how that backbone can form hydrogen bonding interactions with other things. What would those other things likely be? Could be other proteins. Or how about the exact same protein? Okay. If our backbones were long enough, what could potentially happen? I mean, you can even see super flexible people do weird stuff like this.
Fold over.
They can fold over. We could take the top backbone and we could make it touch the bottom backbone, which looks really scary. That's what they usually do in those like scary movies, right? Same thing is happening within our protein. Okay? If our protein gets long enough, that backbone can fold over and interact with itself. It could also interact with the backbone of a separate protein. Or if it's long enough, it could weave around, do a whole bunch of stuff, and then come back and interact with itself. Okay? So what we're looking at with secondary structure is how does the backbone adjust to interact with itself, okay, at some point or another within the structure. There are two primary secondary structures. Okay, and again, these are backbone interactions. We get the alpha helix, which is a relatively short backbone interaction, and we get long interactions, which is a relatively, we get the beta sheet, which is a relatively long interaction between backbones. Okay? So we get a couple different representations. If we look to the center for both of these, so these are kind of split down the middle with the intent to go out that way and out that way. So if we look at the middle, okay, does that look obvious as to what shape we're seeing? All those little balls and sticks and stuff. Everybody looks like, oh yeah, I totally see a helix there. Or that one is totally a sheet. No. Okay, why not? Too many atoms in the way. It gets very confusing. And believe it or not, those are actually simplified because you notice they didn't actually say what the R groups were. They just said that it was an R. Okay? Remember, we had lots of different R groups. If we change those to actually show that structure, it becomes even more confusing. Okay? So what happens is we can then simplify that and say, well, it's secondary structure. All I care about is the backbone. So I'm going to start to remove some of that information. And as we remove some of that information, it starts to clean up a little bit. And when we look at the alpha helix, we see this kind of weird zigzag back and forth. Okay, so, well, that doesn't look very much like a helix. But remember, what is a helix? It's a coil. It's a three-dimensional shape. And this drawing is two-dimensional. Okay, so we run into the same issue we'd run into in, say, organic chemistry and drawing structures. We're trying to represent a three-dimensional shape with two dimensions. And it's not easy to see how that interacts. Okay? Same thing is happening with the beta sheet. Okay? They started to overlay and make this a little bit cleaner because they started to overlay kind of a zigzag shape on it as well. If we compare the zigzags, though, the alpha helix versus the beta sheet, do those look to be the same zigzaggy? No, the alpha helix looks to be really, really zigzagged, and the beta sheet looks relatively tame. Okay? Why? Well, if we scale this all the way out to our fully cartoon animated version, what's happening in the alpha helix? It coils. So it's coming out of the board and back into the board and out of the board and winding around. What's happening with the beta sheet? Okay, it's like a crimp. Right? Yeah, you can do that with hair. You could crimp hair, like your hair's kind of crimped, versus if we did a full coil, like a curlers, right? That's kind of the idea. We've got a crimping, whoops, in the beta sheet, and we've got a full curl on the alpha helix. Okay? Why is it not just straight? The bond angles. Okay? We still have carbon with tetrahedral bond angles, so we will still have that kind of bend to it, but it's pretty much as flat as we can get, okay, without like massively destroying things. What's the head shake?
No.
No, you're good? Okay.
She doesn't like it.
She doesn't like crimping? Okay. Crumping? No, sorry. So we have alpha helix and our beta sheets. Those are our primary structures. So that then brings us to the next point. We want to now look at and evaluate those. And this is where it can get a bit dicey, but we're going to do it anyway. So we've got our alpha helix. We've got a bunch of interactions on it. Okay? So I've got a video for this, because remember, it's a three-dimensional object. If I can just point at stuff and say, well, we get hydrogen bonds stabilizing the alpha helix, because those are those red lines in between, and you can very clearly see where those dotted lines are connecting. Really? You can see that? Jen says no. If you can see that, that's awesome. That's really cool. Okay? One turn of a helix. Anybody even see the helix? Yeah. Okay. But there's one turn of it, and within that one turn, there were 3.6 amino acids. Anybody see that? Yeah. Okay. Not particularly easy. Okay, so I dug around a little bit, and YouTube is full of useful things. and useless things. Clearly, I'm all over it. The alpha, that was kind of a joke. The alpha helix, they got a nice little video here. So we're going to play that video, and then what I want you to consider while watching it is they're going to list off a whole bunch of statistics, like this 3.6, this 5.4, where the hydrogen bonds are, but they'll help you see the hydrogen bonds because what they will end up doing is moving the alpha helix around. So when we see it from different angles, that'll hopefully make it a little bit more obvious what's going on with it. But in the last question there, what conditions might prevent the alpha helix from forming? Okay? That is a challenging question, but that's what we're going to try and consider. And yes, we got to think of at least 3. Interestingly enough, how many of those 3 are mentioned in the video? None. Absolutely none. Yeah. I didn't say I was nice. So I'm going to attempt to do this where potentially the video will show it, but the link is buried in that alpha helix word up at the top. So let's see if this works. Of course it would not work. Stop. Go away. Sorry, apologize for the little delay here. Let's slide that across. If I do that. Okay. So in the recording, if you decide to watch the recording at home or my recording, it's going to look really, really jumpy. And I'll try and do it for the audio. Let's hear the audio too. I apologize if this gets a bit boomy, but we'll see.
Helix is one of the most common secondary structures. Amino acid side chains project outwards from the front of the backbone that forms the core of the helix. The chain is stabilized in this conformation by hydrogen bonds between the backbone amino group of one amino acid and the backbone carbonyl group of another amino acid that is 4 positions away. These interactions do not involve side chains. Thus, many different sequences can adopt a helical structure.
Except
Alpha helices are regular cylindrical structures. One full turn occurs every 3.6 residues and extends the length of the helix by approximately half a nanometer. Secondary structures are often represented in cartoon form to clarify the underlying structure of a protein. In this representation, the twisted 3D ribbon follows the path of the peptide backbone.
Okay, did you guys see anything interesting in there that you wanted to go back and look at? Does it help visualize? You're saying no. Okay, well, I tried. Unfortunately, I don't think I have enough of a model kit to actually fully build that 3-dimensional structure, so all we get are this kind of animation.
So the protein backbone is the green?
That requires going back to the video.
Oh, sorry.
That's quite all right. Okay, so the green there, that green are the R groups.
Okay.
Okay. So what we're looking at, because I think I can do that here. Let's see. Let's try red and see if red will jump out at us. Okay. So our backbone is this kind of. Wow, it's gonna be hard to follow, but I'm kind of attempting to follow it. Whoops, that way. That's our protein backbone. Can you see that? Okay. The green part, which I'll circle in blue, are the R groups coming off of it. Okay, so let's pick. Let's actually try and go through and do it all. So let's clear all that. Whoops. Let's clear all that. Let's look at our first amino acid. Starting up at the top, there's our carbonyl, or carboxylic acid part, going to a carbon, which then has the R group attached to it, which then goes to the nitrogen. Everybody kind of see that? So the blue is supposed to be representing the nitrogen. What amino acid is that? So this gets a bit challenging here. Right there is, that looks like it's a ring, but are my hands touching? Does it look like my hands are touching? Close one of your eyes. If you close one of your eyes, you'll lose your depth perception, and it should look like my hands are potentially touching, until we turn and say, oh no, they're not. Okay? And so we run into that issue with all two-dimensional drawings. Okay, so those actually aren't touching, which would then mean if we go through and colored in our next amino acid, because blue seemed to be working, let's go ahead and do that. There's our carbonyl. There's our carbon where our R group is attached. The R group is now really hard to see because it's going backwards behind all the structures, so the rest of the structure's in the way. And then it goes out to our nitrogen again, a new nitrogen. Kind of sort of see it? I've got it zoomed in on my screen, so it looks really obvious to me. I just can't zoom for you guys. Then we have our purple. There's our next carbonyl, our next R group, and then our next amine. Does that help you kind of see where they are? Okay. So what they're trying to do, or what they tried to do at first, and now I got to get rid of all of that because we need to go back to moving the video, is that they initially, they initially had everything on there. Actually, they full-on drew everything on it first. So there they're trying to highlight the backbone. Okay. Then they started to add information in. Where are the atoms? Red being oxygen, blue being nitrogen. Then they're saying the green is the side chain, so that we can see those extra pieces within it. Okay. And then as they continue, they're just rotating it so that we can see all of those individual pieces, again, trying to bring in some of that three-dimensional characteristic. And let's see, do they... Maybe this is what you were talking... Nope. So that still shows green as the side chains. Okay. What's the face mark? It's just a lot. Okay. And then they're trying to go through and highlight things a little bit different. Okay. Here's an interesting feature. Where are all the R groups? They're all projecting outwards. Okay. Could they potentially project inwards? Where's the image I want? If you look at the alpha helix, the R groups must be projected outwards because the alpha helix is tiny. Okay? There is no space for an R group in the middle. If the R groups were somehow aimed inwards, what happens to the alpha helix? It breaks apart and we no longer have it. That does not destroy the primary structure, that destroys the secondary structure. Okay? Kind of makes sense? Okay. So, and did that get you back on what the greens were to help with that? Okay. And then let's make sure they didn't do something weird to make something else really green. It looks like they were consistent within that. And then they go through and say, well, here's the ribbon showing the backbone. And again, depending on what we're looking at, we may want to know what amino acids are there, so we'll show the R groups. Sometimes we won't. Okay. Typically, what you end up seeing is something along this line. And we end up not showing the R groups. Okay, because we're focusing on the secondary structure.
So when you say backbone, you mean like carboxylic acid?
So, careful.
Or amino acid? Is that what
Yes.
Okay.
It's what makes the amino acid. So that if we were looking, well, I'm here. Let's just do it on this. Okay. When we're looking at our backbone, this was an amino acid. Oh, that was awful. I apologize. Let me try that again. Okay, that is an amino acid. That is our R group coming off of it. The backbone of our amino acid is really just referring to those atoms, not the OH, because the OH is not an R group. It goes away when I make the protein. It goes away with, whoops, with the one H that is also not highlighted in it. Okay? Those get lost in what we call the condensation reaction, because in the condensation we lose water. OH and H is our water. So the backbone is just referring to the atoms that are identical for every amino acid. Does that make sense? Okay. Let's see if I can get back to this. Other questions on the video? You can always ask and I'll go back, but I won't move it this time.
So quick question, when you were asking what were the 3 amino acids in that,
Oh, okay, yes.
there was, back when they were kind of colored green earlier in that video,
Mm-hmm.
how do you, with that top one, how do you differentiate right there? When you were highlighting that top one, how do you differentiate, because it's like, is that glycine or alanine?
Ooh, interesting question.
Because one is just hydrogen is the R group, the other is a CH3, so I know it's kind of weird, but how
Yeah.
do you differentiate
If
without knowing?
we looked at the backbone of our chain, right? We had, oh, I'm going to draw it again and then delete it again. Whoops. We had our backbone. We had the, come on, pen. We said that was our amino acid, and our backbone was this, right? And that was our R group. Okay. What atoms are in that? Carbon, hydrogen, carbon, oxygen, carbon, nitrogen, right? What atoms are shown, or what bonds are shown in this backbone? Carbon, 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 nitrogen, carbon, oxygen. Where are the carbon hydrogens? They're implied. So this goes back to organic chemistry drawings. We'll imply the carbon hydrogens. So that green one at the very first, yes, there's something there, but if it was hydrogen, it would be implied. So I wouldn't show anything. So that's probably why they didn't pick glycine. Glycine, thank you. Okay. Because they wouldn't be able to highlight anything. They'd be like, well, what do I do? So they picked alanine because they could actually highlight that. Does that make sense? Okay. Other questions? Okay. What conditions might prevent the alpha helix from forming? There are 3. And actually, since I don't like that picture, I'm going to see if I can find a better picture in the video. Okay, but think about it. Or while I'm trying to find a better picture, think about it.
You say it has nothing to do with R groups?
I did not say that.
Okay, then.
The secondary structure is due to the backbone and the backbone only. But do the R groups potentially affect what the backbone can do? Yes.
Is it the size of the R groups, if they're on the same side, could repulsion, or the charges, if they're opposite or the same charge, opposite but repulsive, or
If
it's the same?
we had R groups here that were big, massive, sterically big things, what happens when they get near each other? They repel. If they repel, what happens to the alpha helix? It starts to bend or spread. If it starts to spread, it's no longer an alpha helix, we lose that structure. So our R groups being too large and repelling away from each other through a hydrophobic interaction, right? Or even hydrophilic, I mean, just we're saying large, okay? That can prevent the alpha helix. Okay? Which you then start to suggest the next logical step from that. What if they were ionic? Do we have charged R groups?
Yes.
Yeah. What if we had 2 R groups that were the same charge? They're going to repel away from each other and cause that exact same issue. Okay? So good, that's 2. Could do a really unfortunate and almost useless hint. We could say, which amino acids did they show in this?
Phenylalanine, alanine, and choline. First three.
Okay, so we had, what, alanine. That looks like probably threonine, I think is that one. We've got what looks to be histidine. We've got, maybe that one's threonine and that's isoleucine. Okay. Trying to nail those down is going to be easy because we don't have a lot of information. We're just saying how many atoms are in that. Okay. But that's again getting us to think about the actual amino acids. Okay. Well, all of those amino acids should appear in, or are allowed in, alpha helices. That's why they showed them. Maybe our answer, our third answer, is what amino acids did they not show? Okay, and unfortunately, there are not 19 things there. Okay, so that narrows our focus a little bit, but it might, with that hint, narrow it enough that you can pick up what we need. So I'll write down the two things we can get. Our group, we'll call it hydrophobic repulsion. That says repulsion. And then we have our group, electrostatic. So that's our similar charges. Repulsion. Then we have a 3. Okay? And the hint we gave is what amino acid not shown in the video. Nice big vague hint. Glycine actually won't be a problem. Anybody have, well, I think Mark has it at least, the list of all the amino acids? And potentially, because we're looking at structural effects, we probably want to know not their 3-letter codes or 1-letter codes or names, but want to look at the structure. Is there an amino acid that limits how the backbone can be adjusted? Proline. The R group in proline comes off the standard carbon and then does what? Comes right back down and bonds to the nitrogen. What does that do to the bond angle between the carbon and the nitrogen of the backbone? Not only does it change it, but it forces it to be only one angle. It can't do anything else because it's now locked into a ring. I can't do rotation there. Okay? To get the proper rotation for an alpha helix, proline prevents it. Proline will not allow for alpha helices, or at least nice alpha helices. Okay? Proline is really, really bad. Okay? So those are the 3 primary characteristics that will mess with our alpha helix. Okay? Because we've got a test coming up, we might want to be like, well, what are we really going to get tested on? Okay? I do not expect you to memorize how many residues are in a turn or the distance between things. I do expect you to know that hydrogen bonds stabilize the alpha helix, and I do expect you to know that those hydrogen bonds occur where? I don't accept between structures. Too vague. In between what? Amino acids is too close, or too vague as well. If we look at the shorthand, there's our shorthand, the nice ribbon structure. What is missing from that ribbon structure? The R groups. What makes the alpha helix? Hydrogen bonding. If it's within the R groups, then I had better show them on my structure. It's in the backbone.
But isn't that between the amino acids?
Well, so the amino acids was too vague because it is between the amino acids in the backbone, but if we said the R groups, that's between amino acids too. Right? It's It's not between the R groups of our amino acids, it's between the backbone of our amino acids. Okay, so saying amino acids is too vague of a term, I can't nail down what you're referencing. Okay, so that is an important thing to distinguish. The other thing, the conditions, the answers to that question. Okay, the conditions that prevent the alpha helix from forming, that will be important. Okay, notice the things I'm telling you to not worry about all involve numbers. Okay, because I can't count. Okay, kind of makes sense? Okay, so there's a big list of helix facts. Is that word? Okay. Most of those are not super concerning. Okay. We could make some kind of references. 7, that's going to be an important one. Where are the R groups? All have to go outwards. Those of you being like, where is this on the slides? It is on that slide. It's just buried underneath something that you can't see. Okay. So these facts aren't within the slides. So the R groups facing outwards, that is an important fact that needs to show up, okay, in addition to the ones we listed. The hydrogen bonding being parallel to the coil axis. Less important, but if we look at it, our coil axis is running up and down. Where are all the hydrogen bonds? Running up and down. Okay? So it's kind of an inherent one. It's just kind of cleanly stated there. Amino acid bonded to the 4th amino acid that is distant. For those of you taking like MCAT level tests, I think they start to get nitpicky like that. I don't care. The amino acids being trans and planar, don't worry about. We just said we don't worry about those. The clockwise right-handed is kind of a neat, important fact. I don't think I would ever test on it because I don't have that memorized. Okay. Could we have counterclockwise left-handed? Theoretically, why is it only clockwise right-handed? What things or have we seen this whole right versus left thing pop up before? Chirality and stereocenters. Our amino acids all have a stereocenter and they're all consistent, okay? Which then is going to force any kind of chirality in anything that uses them. That's why we're seeing the clockwise right-handed. Okay? So it comes back to the amino acids being used. Because the amino acids have a chirality, they force that chirality on their secondary structures. Okay? So kind of a neat fact. That might be me being kind of a jerk asking that question, but I kind of doubt I would ask that. So I'll just do kind of a dotted X through that one. Okay? Beta sheets. Okay? What's happening in the beta sheet? We're taking strands of our protein and we're matching them up with each other. Remember, our protein backbone has lots of places to hydrogen bond. If I take a chain of protein, right, well, there's lots of places. I could then line that chain. Man, excuse me. Okay, I could line another chain next to it. Okay. That interaction, if it's strong enough, is now maintained, and I would get the beta sheeting. I would get the crimping effect, as opposed to just letting the hair or your proteins just flop all over. Okay? There's 2 formats to this. We can run parallel strands or we can run anti-parallel strands. Okay? Because remember, our amino acids or our proteins have a direction. That direction runs amine to acid. Okay? So depending on which direction those strands align, either amine to acid or acid to amine, I can get parallel or anti-parallel sheets. And we got an image for that in a second. What happens to the R groups? Well, R groups are coming off the backbone, so if this is my protein and this is my protein, if the backbone has to interact, where do the R groups have to be?
They get rotated.
Okay. So we could say to the side, but couldn't I just as easily do one over here? Okay, so you can't say it's to the side.
Front to back.
Up or down. So depending on which strand we've got, the R groups will get forced either up or down because that's the only direction that we can't build our beta sheet within. Okay? They also maintain this S-trans planar. Since I didn't talk about the S-trans earlier, we'll just kind of ignore that statement. Okay? And so your carbonyl and your nitrogen are perpendicular to the axis of the sheet. So if my hands, straight arms going out, are the axis of my sheet, where are the carbonyls and the nitrogens? Where's perpendicular? Yes. Where my thumbs now are. We point the thumbs inwards. That is now perpendicular to the axis because they're running this direction, yet the sheet is running this direction. How that make sense? Okay? So, our parallel versus anti-parallel. Okay, in the parallel case, what's happening? They're running in the same direction. We're going amine to carboxyl, amine to carboxyl. Okay, so that's what's happening in that top case. Is that obvious based on how it's drawn? Okay, for those of you saying, yeah, I can see the arrow. Ignore the arrow. I would argue with based on how that's drawn, that's not super inherently obvious because they didn't start it very cleanly at the amine, okay, or the acid. It's kind of confusing in looking at that. Okay, that also means I'd have to draw a lot of structures, and so I probably wouldn't ask that either. Okay. If we look at the anti-parallel, what's happening? Okay, they're going in the reverse direction. So we have parallel versus anti-parallel. Okay. How could I possibly get parallel sheets or parallel protein strands? How do I make my arm, there's my protein strand, how do I make that a parallel sheet? What's that? What do I rotate? Just kind of rip that off, turn it around, and line it up. So that's kind of problematic, right? Mm-hmm. Okay. How could I get anti-parallel? Boop. There it is. My hand is now anti-parallel to my forearm. There's a beta sheet that kind of hurts. Okay. How do I get the parallel strands? Can I do it really easily with this one hand or one arm? Okay. I'd have to rip the arm off, put it down there so that it runs parallel. How else could I do it? Bring in a second one. There they are. Now they're running the same direction. Okay. What does that mean about the distance between those strands? Right? And I don't mean in the beta sheet. I mean when I leave one amino acid and make it to the next amino acid in the strand. If they're running parallel, what does that mean? Can I possibly link this back to that? Why not? If this is going nitrogen to carbon, 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 is that running the correct direction? No. How do I get that top strand to match that strand? What do I have in all of that? I'm not even showing that interact. I'm just saying, what's out there? How do I get that to happen? What do I need? A lot of amino acids. I need a big amino acid loop to somehow come back around and line back up. What about for the bottom one? Okay, I'm going to have to start the bottom one from the very bottom. Nitrogen to carbon. So we're going that direction, right? How can I get it to loop to the next one? Whoop. What's happening here versus up in the top? The purple one is happening almost an instant U-turn. What's happening in the red one? Really big, and I would argue that's probably not even a U-turn. Okay, what is it? More like a hairpin? Okay, because you're flipping back around and then back a line. You've got almost 2 U-turns into it. Okay. This brings in kind of a characteristic of what we can see. Whoa! Come back here. in our antiparallel sheets, what amino acids would we expect to see in that U-turn? Why proline? What does proline do to the backbone? It makes a ring, and it has to force it in really, really tight. So if you drop in proline as one of those next amino acids, okay, we can force that bend relatively quickly to get that to come back. What amino acid might we expect to not find in that sharp U-turn? What's that?
One of the bigger ones,
Why one of the bigger ones?
it won't be as, well, there'd be rotation, so it's not forced to go in that direction.
What's the issue with big ones?
Stir, strain.
Steric strain. What are the steric strain going to do? They're going to repel away from each other. The whole point here is to do what? A quick turn, which means little space or no space, little space or a lot of space. Little space, which means large amino acids are going to be a problem. Okay. I could use a bunch of small amino acids. They won't occupy a lot of space. They won't block each other, and I can spin that corner a little bit faster. Okay? Yes?
I don't get, like, why it says that it goes from nitrogen to carbon, because it starts out with,
Yeah, the image there is stupid setup on this. The image drawn is pretty horrible. What they're trying to do is show that you've got a longer protein structure, but we're only looking at one section of it. Okay? So if we go through and evaluate, if we're saying that that is our carbonyl, okay, what's the issue? If that was then my protein, the carbonyl, my individual amino acids go from the carbonyl to what? The carbonyl is not connected to the nitrogen in an amino acid. It's connected to a carbon. Okay? So what are we showing in that linkage right there? That's not one amino acid. That is two amino acids. I have one that's coming off and going, or part of that page, and then I have the next amino acid here. And then I have the next amino acid here. Well, let's just kind of do it as big U's. Maybe that'll show up a little bit nicer. And I have the next amino acid there. And the next amino acid there. Do you see those? Okay. Look at an individual amino acid now. Let's look at the red one right in the middle. Where's the nitrogen? And the carbonyl? Look at the purple one. Where's the nitrogen? Carbonyl? Left. Showing the direction as they've got shown. Okay. If we move to the bottom one, okay, they're again showing that nitrogen. So maybe they're saying that that's the first amino acid. Is that the structure for an amino acid? No, unfortunately, again, they did the same thing. There's an amino acid. There's an amino acid. There's an amino acid. For that purple one, where's the nitrogen? On the right. And the carbonyl on the left. For the green one, on the right, on the left, that matches the direction they're showing here. Okay. Does that make sense? So be careful looking at a chain and just saying it's starting with the first amino acid. Rarely that's true. Okay. So draw it out, find where an amino acid is, and then what I would do is highlight a couple of them, like I just did here. And then say, okay, where's the nitrogen, where's the carbonyl, which direction does that go? Now the next one, where's the nitrogen, where's the carbonyl, which direction did that go?
so
Okay.
parallel always have nitrogen on the right.
I could have picked this up, though, and turned it that direction. Okay, so we can't just say always on the right or always on the left, which hopefully would have been discussed in organic chemistry. Okay, because the example that I like to use, which Jen is probably sick of hearing, well, what's my name? Uh-oh.
Is that a trick question?
No. What is it now? Would you have drawn me differently, though? Yeah. Same thing's happening with our structures. Okay. The name is still the same, but we're looking at it from a different angle. So be very careful to say it's always this direction, because it won't always be that direction. We can flip and look at the structure from a different angle. Okay? So that, again, comes back to why I would identify my amino acids, that sequence, that piece, draw it in, say nitrogen, carbon. Okay, that way, nitrogen, carbon. Okay? What happens, let's say we went through and did that. Let's clear that all up. Let's say we looked at this one, and we looked at this one. So red and purple. Let's say, just for the sake of argument, so ignore what it actually says there. For the red one, we said it was going this direction, nitrogen to carbon. And for the purple one, we saw nitrogen to carbon. Okay, so there's a couple big answers that can come out of that. Okay, Mark, you shook your head no. Why did you shake your head no?
Because that's not consistent with the backbone.
That's not consistent with the backbone, meaning a peptide bond wouldn't form, meaning we don't have amino acids. Okay? Biochem, what should we be looking at? Amino acids. So what does that mean? Whoever drew it made a mistake. That's when you raise your hand and be like, dude, you screwed up. That's fine. I'll be like, yeah, I did. Let's just ignore that question. Pretend it didn't happen. Okay? Or I'll be like, yeah, the book screwed up. Good eyes. Okay? But when we're looking at amino acids, they will hold that same pattern. They will never contrast in an amino acid sequence. Okay? So, secondary plus structures. So, what we start to move, so if we had primary and we had secondary, what do you expect to be next? Tertiary, and followed by quaternary. Kind of like we did with organic chemistry. Primary carbons, secondary carbons, tertiary carbons, quaternary carbons. Okay. We're doing a similar thing within biochemistry, but just kind of scaling up the difficulty. Remember, primary was just a straight chain. Secondary said, how did the backbone of that chain interact to change our view of the molecule or our view of the protein? Okay. Tertiary is then going to be the next step up. But before we move to the next step up, we run into this weird kind of gray area that I'm calling secondary plus. Okay. Someone went through and looked at a bunch of proteins and said, hey, that looks like a coil. Let's call it an alpha helix. It didn't make up the whole protein. It was a part of it. Okay. Someone went through and saw, look, those strands are all aiming in the same direction. That looks like a sheet. Okay. I'm going to call that a beta sheet. Why did they call it beta versus alpha? Which do you think they discovered first? Alpha. Okay. Beta, because it was the second secondary structure discovered. Okay. Then somebody went through or found more proteins. We were able to isolate them and look at their structures in enough detail that we started to notice these other things. We noticed this thing, where the beta sheet was there, but then what did the beta sheet do? Because those lines represented our beta sheet. Are those parallel or anti-parallel? Anti-parallel. Okay, is it just a beta sheet? When you look at your sheet, right, it's this nice flat thing. Is that nice and flat? What's it doing? It's coiling back on itself. It's making a barrel. It typically gets referred to as the
Beta barrel.
Beta barrel. Because it's made up of a beta sheet that is coiled back on itself, and we have a beta barrel. Okay. Is that secondary structure? Well, what makes it is the backbone interactions, and those backbone interactions happen to be happening for a long enough distance that it somehow coils back on itself to make another structure. Well, should I call that a gamma barrel? Alpha, beta, gamma, right? Or is it delta? Whatever. The next Greek letter? Okay. Why? It's still doing the beta formation. Okay. So we'll call it a beta barrel. Okay. So we can get those kind of anti-parallel. We can get more anti-parallel. Okay. And then someone was like, okay, well, this one's a little bit smaller than this one, so we can give it a new name. Why? Because someone wants to be famous for naming something. Really. Okay. What happens to the next one? I have alpha helices coiled on top of a beta barrel, which was made up of a beta sheet. That's not the entire protein, though. It's only a small section of it. So our secondary plus is starting to look at how some of our kind of primary, secondary, or our principal secondary structures can interact with each other to form larger structures, but not being the entire protein. Because the entire protein is known as the tertiary structure. So we're kind of in this gray area on what's happening, and that's why I refer to it as a secondary plus. Okay? Because it's still not the entire protein. Okay? It is a section of it, and it's still really happening due to the backbone interactions. So that really means it's secondary, but it's a little bit more advanced than just simple interactions. Okay? So we end up with a bunch of other things. These typically get classified as motifs. Okay? And so they'll come up with weird names for different structural motifs. Alpha helices and beta sheets are motifs. They just happen to be the most common ones that you're forced to memorize. Okay? The other ones you aren't forced to memorize, but they show up kind of frequently depending on what we're talking about. Okay? So you can hear of zinc fingers and things like that. Okay? A lot of those are different secondary structures that are less common, okay? Which are probably not quite as less common, but not as well studied. So we can't force you to memorize them as cleanly. That make sense? Okay. So as we're transitioning between that secondary and tertiary, we have our common repeating motifs. In our secondary, primarily our alpha and our alpha helix and our beta sheets. Okay? When we move into tertiary, it's then how do those individual motifs interact with each other? Okay? And it can be a step more than that. Because if we just say how the motifs interact with each other, we're ignoring the rest of the protein. Tertiary structure says how the entire thing comes together to make the protein. Okay? So we end up with two primary categories when we're looking at these proteins and how they then interact for a larger structure. We end up with what are known as fibrous proteins, or at least that's how I say that, and globular proteins. Okay? Our fibrous proteins, collagen, fibroin, keratin, globular proteins, because we've talked about it a bunch, ATP synthase. Okay? If we think about fibrous, think about a fiber. What do you think about when you envision a fiber? Strands. Okay, long strands. Okay, I wonder why I use strand as a word. Okay, globular proteins, globs. Globs, you're thinking kind of balls, amorphous shapes. Okay, if we're thinking about solubility, what would we expect for solubility for each of these? What do you mean not? What's not? This one or that one? Okay, interestingly enough, if they're not soluble, what explains all of us? We'd have to have a third category, right? To explain everything else that was happening in the water environment. There's only two categories, which means one of them might not be and the other one might be. So which is which? Globular ends up being soluble. Okay. Fibrous are not soluble. Okay. Have you heard of collagen, fibroin, or keratin before?
Yes.
Okay. What are they used for? Tissue. Okay. To do what with the tissue? Make it stronger. Okay. If it was soluble in water, what happens? It would fall apart because we're 80% water. Okay. So our fibrous proteins are not very soluble in water intentionally because they're trying to make structure. Okay? If they weren't there to provide structure, then they would just fall apart. The globular proteins, on the other hand, tend to be very soluble in water. Those tend to be the types of proteins that will move things around or cause reactions to occur in the environment, okay, or in a water environment. Okay? They're a lot more mobile. The fibrous proteins tend to be a lot more locational and structural. Okay? Which then gets us to the cell role. Cell role for our fibrous, just said it. Structure. Okay. Cell role for our globular proteins, processing. Okay. This does not mean there's no crossovers. There are crossovers, but those are general rules or general categories. That's what sets them apart. Okay? Tertiary structure. Okay? Tertiary structure. Did I miss something, or you guys got it?
No.
Okay. Tertiary structure is very, very complex. We get lots of different things happening within it. So we've got a whole bunch of stuff shown down here trying to identify it. What's the first thing you see? Alpha helix. Thank you. Awesome. Alpha helix is our secondary structure. Why are we showing it in the tertiary structure? How does the alpha helix interact with the rest of the protein? If it's interacting with the rest of the protein, that's going into tertiary structure. What part of the alpha helix do you think would interact with the rest of the structure? R groups. Okay? Which then by definition means we can't be secondary because the secondary is looking at our backbone. Why would we not expect the backbone of our alpha helix to interact with anything?
Because it would break up.
It's coiled inside, so yeah, you're right. It would break up because for it to interact, it would have to come from the inside out. Okay? We have our kind of R groups acting as almost a shield or a sheath on the outside of it. Okay? We have our beta sheet, which is what they're attempting to show with those big solid lines, which look like bonds. The issue with that, they're not actually bonds, even though we call them hydrogen bonds. Okay? So to add to the confusion that you were asking about earlier, yeah, now we start showing them and making them look like hydrogen bonds. So other things that can contribute. M plus 2, what is an M? It's our generic term for a metal. Okay? It does not necessarily have to be plus 2, but the metals that contribute to structure typically are plus 2 in charge. Okay? If we take a look through here, isoleucine, valine, leucine, what can you tell me about those 3 amino acids? I don't accept that they're smaller. They're actually medium to large. They're all hydrophobic. They're your nonpolar amino acids. What are the nonpolar amino acids going to do? Avoid water. Where is water with respect to this protein? Everywhere on the outside. Where are they showing those amino acids? On the inside. Okay? They're mentioning disulfide bonds. Okay? So we can get sulfur. Sulfur is a nasty molecule because it can do all sorts of weird chemistry that just kind of messes with organic chemists and it messes with biochemists, except they ignore most of it. Okay? And we can link sulfurs across an amino acid sequence or a protein sequence. And we can form a physical bond connecting them. That's kind of neat. That physical bond is easily reversible or relatively easily reversible, okay? Which is why we refer to it as our tertiary structure. We can get hydrogen bonding between the side chains. How is that different from secondary structure? Secondary structure is between the backbone. Okay? The side chains, hydrogen bonding, contributes to tertiary, not secondary. Okay? For those of you answering homework questions, there's a very subtle, nasty question involving that. Okay? That maybe you'll get lucky and avoid. And then you also get electrostatic interactions. We have side chains that are charged. Those charged side chains can interact with each other, okay, to form strong interactions, showing here a positive nitrogen and the negative carboxyl. Okay? So we've got that big list, which then also would beg the question, which is one of the reasons why in biochem you're typically expected to memorize the amino acids. Which amino acids would we expect to contribute to each of these things? What was that? Okay. Glutamic acid or glutamate. It would actually be glutamate at physiological pH. And where is that going to show up? Which one of those interactions? Okay, we would likely see that being glutamate. Right? Okay. What else?
Aspartate.
Okay, aspartate could show up there. Was that ASP? What other amino acids could pop up in
Lysine.
anywhere in this chart?
Oh.
When you're saying glycine?
Lysine.
Okay, lysine for, okay, I think I'm thinking of leucine. Lysine's the NH3? We have lysine potentially interacting there. Okay, that's our NH3. Arginine was the one I was thinking of. Okay, histidine could go in there. The disulfide bonds are going to be due to cysteine. Okay, what other amino acid could contribute to the disulfide bonds? What's the issue with methionine? Methionine is nonpolar. It has carbons on both ends of the sulfur. If you have carbons on both ends of the sulfur, you can't make two bonds. Only cysteine can do your disulfide bonds. Okay? So being able to take the amino acids that you've memorized, your 5 amino acids, and subdivide them out into, okay, where would I expect to find those, is an important thing to do. Okay? That's an important aspect of looking at biochemistry and protein structure. Kind of make sense? Okay. Actually, sure. Not quite yet. So, protein folding. Okay. What do we see as a difference between these? I know we're almost done. I know it's not super obvious, but see what you can come up with. What are we highlighting as a difference between these two? The side chains. Where's that difference?
On the right side.
Right side is what? Highlighting which side chains?
They're highlighted in green.
Green. And on the left? Reddy orange. Okay. Where are the green ones? Where are the orange ones? What can you tell me about the orange ones versus the green ones? Don't tell me outside and inside. Smaller's not a bad idea, but not where we're going with that, because it doesn't necessarily have to be smaller to be on the inside. Polarity, are polar and nonpolar, or hydrophilic, hydrophobic. Okay. The red ones, or the orange ones, however we want to look at that, would likely be our nonpolar. And our green ones would end up being our polar or hydrophilic ones. Okay? So I kind of got to end there, so I'll stop that.